Assalamualaikum and hi everyone. My name is Nur Aishafika binti Mazuki. I'm from DBT 3A. My metric number is 23DBT20F 2011. Okay, today I want talk about SDS Page. Okay, the theory behind the sodium dodecyl sulfate polyacrylamide gel electrophoresis, also known as SDS Page, is to separate protein based on size through the use of stacking gel and resolving gel. Okay, the sample that will be observed are loaded into the well after being combined with a buffer containing 30% glycerol, SDS, and thiol. Uh, Let me show you the stimulation of SDS page. Okay, these are the material needed for this experiment. As you can see, we have 6 steps. Okay, the first step for this experiment is increase the heat block temperature to 65 degrees Celsius. Okay, after that, set a micropipette to 30 UL and attach a tip and save a column after you have done. After you have done, attach a tip, and after that, draw up the Y type of cell culture. After that, close the Y-type cell culture and dispense the Y-type cell culture into the Y-type cells tube. And then eject the tip into the biohazard waste container and repeat the procedure to transfer 30 UL of the Baker cell culture. The same step like pancreas cell, we also repeat the procedure to transfer 30 UL of the edit 4 cell culture and repeat the procedure to transfer 30 UL of the edit 7 cell culture. Okay, next step is set a micropipette to 10 UL and attach tip. Before that, after you set 10 UL, you must save the volume. Okay, after that, open box and attach a tip. Draw up 10 UL of 4x SDS sample buffer and close the 4x SDS sample buffer. After that, dispense the 4x SDS sample buffer into the white type cell tube. After that, you must repeat the procedure to dispense 10 UL of 4x SDS sample buffer into the pancreas cell tube. Also, also, repeat the procedure to dispense 10 UL of 4SDS sample buffer into the edit 4 tube. Okay, lastly, repeat the procedure into the dispense 10 UL of 4S SDS sample buffer into the edit 7 tube. The protein ladder already contains the SDS sample buffer. Next step is move all of your 4 sample tubes to the heat block. After that, set the heat block timer to 10 minutes and after that press start. After 10 minutes done, remove all of your sample tubes from the heat block carefully. Next step is move all of your sample tubes to the microcentrifuge. Transfer it carefully, and after that you must balance the microcentrifuge like this. After that, adjust microcentrifuge control, and after it's done, you can remove the tube carefully. Okay, next step is you must place the polyacrylamide gel in the electrophoresis box. Place the polyacrylamide carefully. After that, fill the gate cassette with 1x running buffer and remove the comb. Set a micropipette to 20 UL and attach a tip. Before that, you must open the box to attach a tip. After that, draw up the protein ladder carefully and move the micropipette to the first lane of the protein gel like this. You can see in this video stimulation. This step you will be repeat again and again, but with different cell, like cell of tube, pancreas tube. After that, dispense 20 UL of the negative control into the third well of the gel, like this. And then dispense 20 UL of the edit fossil into the fourth well of the gel. And lastly, dispense 20 UL of the edit 7 cells into the fifth well of the gel. Okay, the last step is cover the electropolysis box and connect the red and black cable to the power supply and the lid of the gel electropolysis box. Handle it with carefully. After that, you must turn on the power supply, and then set the voltage to 150 voltage and set a timer to 60 minutes, and then press start to the power supply. Wait until the time has elapsed. After that, turn off the power switch and unplug the electrode. Lastly, inspect your gel. This is the result for this experiment. As you can see, this result. Thank you for watching. Begin until the end.